Welcome to School Report, a glimpse into the classrooms of St. Lucie County Public Schools. School Report provides the opportunity to share with the community academic and extracurricular activities of students in our schools. And now, here is School Report.
So this is the lab for the crabby crustaceans in marine science. And so what we're going to do is we're going to take advantage of the fact that we have some shellfish here at the school. And what you're going to want to do first is measure in centimeters from the tip to the tail, okay?
5 inches, flip it around. There we go. So sitting 13.
13, okay, good. We'll remember that number. And so what they wanted us to notice too was that there's jointed appendages, and these are sensory antennae. And from this one we could tell that it's female because of the eggs in there, that it's ready to lay. Okay, so that's pretty much what you need to do for this lab. And then you just write up a little bit of the questions, and then you're all set. So, very good. Well done.
Thank you. I learned so much today.
Okay. What she's demonstrating is what we call agglutination, and so what she's going to do is add a couple of drops. It's fake blood made with milk and red food coloring, and then when antigens are present on blood cells and they're exposed to antibodies, they do what we call clumping. So she's going to determine whether or not an antigen is there by pouring the antibody on and then mixing it. This is a protocol I got from the internet. I was going to try to cite it specifically, but I don't have the citation with me today. Okay, so what we see here is that the blood is clumping. It's, like I said, the fake blood is clumping. So that tells us the antigen is present. And in B, we do not see clumping. So that tells us the antigen is not present. And for the RH factor, it is clumping. And so what we know from this particular blood sample is that it's an A positive blood type.
That's okay. Do you want me to look at it?
Oh yeah, look at that, holding it up.
I don't want it to fall.
Okay. Awesome, good job. And we're going to continue talking about Mr. Mendel and his garden peas. Remember where he crossed the two different flowers and he came up with these different characteristics? Okay, so this is the concept we're still working on. So these were the things that he taught us. Traits are controlled by 2 genes, one from each parent. And then some genes are dominant. 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 And some genes are right. So he also taught us there's opportunities for genes to mix and match, like the crossover that we learned about, and also independent assortment. And that's basically how you don't look like your brothers and sisters. Exactly. So we're going to review some of the different things that we'll look at. This is a karyotype. This is what we see when we line up the chromosomes side by side. Humans have 46 chromosomes, and they come in homologous pairs. And so one came from mom and one came from dad. And then down here is the gender chromosome. So if you have an XX, you're a female, XY is male. So let's talk about some boys, okay? Here's the thing with the boys. You have an X chromosome and a Y chromosome. They look the same? No. And so what that means is they don't have two copies of the same information. Okay? So let's say this particular boy has the gene for color blindness. Okay? It's a done deal. The boy would be colorblind. Now, if a female had the gene for colorblind, she has another X chromosome that might cancel it out because not colorblind is the dominant. Does that make sense? So it's more likely that you're going to have a colorblind male than a colorblind female. Okay? And again, this is the genotype and phenotype concept. So the capital B's, these would be the dominant, which is not colorblind. I'm sorry, the slide ran off. I didn't expect that to happen. But so the dominant is to not be colorblind. You can have, for example, the X with one not colorblind and one recessive. You still end up being not colorblind, right? This is the phenotype. This is the genotype. Okay? And this is what we just talked about with blood typing. In this case, you have co-dominance. In the A-type blood and the B-type blood, they're co-dominant. Okay, so again, we talked about molecules called antigens being on the cell surface of blood cells. And that's what we just saw with the antibodies reacting to it. So if you have somebody that mom has A-type, dad gives B-type, that person is going to be AB. The baby will be AB. Those are co-dominant. If mom gives A and dad gives an O, they still have type A. So it takes two O's to make type O. And then the positive and negative is just the Rh factor. You either have it or you don't. Okay? So here's some traits that we're going to look for today in our exercise. Like I said, when they actually do karyotyping, they're generally looking for disease. Okay, people don't just do the amniocentesis just to find out if their child's going to have hitchhiker's thumb. That's not why you do an amniocentesis. What you do is you look for disease. But I mean, let's not be scary and think about all the negative things. Let's just look for stuff we know about. So the peak hairline, that's when the hair comes into a point on the forehead. Some people have that. Sometimes it's called widow's peak. Freckles, I have that. Dimples, a cleft chin, that's like the little notch there. Christopher Reeves has it. Superman has it. Hitchhiker's thumb, that's where we all stuck up our thumb. Where's my hitchhiker thumb? Oh, there's my hitchhiker thumb. You want to hold it up for them or not? Okay, and then earlobes. You might have free hanging earlobes or you might have attached earlobes. Okay, so these are some of the things we're going to look at. We're going to look at also the case where in mitosis, remember this is back to mitosis. It's all coming together, right? It's coming together. Okay, here you have the metaphase, the chromosomes are lined up, and here you have anaphase separating them. Well, here's the case where one of the chromosomes went to the wrong side. And so now you're going to end up with 3 copies of that chromosome. And that's basically what Down syndrome is. It's 3 copies of chromosome number 21. And so you can see that in here too. That extra chromosome stays there. Sometimes they call it trisomy 21. So Down syndrome is also called trisomy. Tri is 3. Sickle cell anemia is something that we'll look at in our karyotype because it's really common. You know, I wanted to give at least one example of a disease. So what it means is that the blood cell, instead of being this nice disc shape, has a curve to it. And it can be a problem because it can plug up veins and arteries because of its shape. Okay, but this is a lot predominantly found in Africa. In Africa, it's not bad to have sickle cell because you don't get malaria. So it's really a question of perspective, right? It's not good because you get some clogging, but if you're in an area where malaria is prevalent, it's actually an advantage to have this disease. Okay, and then another one we'll look at is the cystic fibrosis that you saw in the video. where there tends to be a lot of mucus in the lungs and the body can't clear it, and so then that causes a lot of bacterial infection. So we're going to take a look at situations of that as well. So again, we'll see that notation dominant will be capital and recessive will be lowercase. And we also remember that dominant doesn't mean more prevalent. Okay? So just because this is a C, the dominant is to have no cystic fibrosis. Okay? And that's going to be on your little sheets that you fill out to help you. Okay, so I think we're ready to do that assignment. What we're going to do is we have bags of chromosomes. You have a set of chromosomes, and you're going to line them up just like that. So if you would pick out a color from over on this table, just make sure we get one of each. Okay, each person will get a set to line up.
I call pink.
Did we get a yellow? Did we get a yellow? Did we get an orange? You got the yellow. Where's the orange? I'm going to switch out your pink because she called pink. Okay, so what I want you to do is take your chromosomes out and you're going to line them side by side so they're numbered. You'll know who's who because they're numbered on the bottom. Okay? That comes from Carolina Science. you're going to have to figure out if you've got an XX or XY. So you'll have to find out. We'll have to go through the list and figure out what you got. Okay, so what you want to do is, what I did is I gave you 4 different tables, so you can read yours, and then you can go to 3 other people's and read theirs as well, but it'll already be set up, which is nice.
Oh.
Okay, here's a 9. 22, here's a 22. Oh, you're going to be hunting. You're going to be hunting chromosomes.
I'm missing a Y chromosome.
Are you?
Yeah, it's nowhere to be found.
Well, we'll have to see why that is. This is a match.
Yeah, I'm missing 2.
Here's a match, and here's a match. Okay. Alrighty, so if you take out your little chart, did I just take yours? You have one?
Yeah.
What? You have one? Where's yours? Oh, okay. So if you take out your chart,
Probably men.
let's determine, first of all, on the top, you can write your kit color. So if you're orange, light blue, pink, green, let's write that down because later on you can check your answers. Okay, and the next thing we're going to do is determine, do you have a boy or a girl?
A girl.
So if you have 2 X's, you've got the girl. If you've got an XY, you've got the boy. So are you missing chromosomes? Are you really still missing a chromosome?
Oh, no. Just the Y.
But you're not a boy, so you're good. You only need 2 of those. It just happens to be the same. Okay, so let's look at chromosome number 9, and let's look at our blood type. Remember, A and B are codominant, O is recessive. So, like for example, where's yours? You've got 2 O's, so that's pretty easy. What's your blood type?
Oh.
O. So what you want to do is you want to write in the genotype, I-O. It's off to work we go, I'm sorry. I-O, and then your phenotype is O. Okay, and then in yours, because remember that's like what's expressed, phenotype, right? Yours yours is good too. You've got 2 A's, so that's a nice, easy one. And then let's see, yours, did you find chromosome 9? So you have I-O as well. And you have, where's your 9? There it is. You have IA. So you got an A. What's yours? You have IBIO. So what's your, perfect. So she wrote for her genotype IBIO, and then the phenotype is B. That's perfect. How about you? IBIO. Okay, B. Perfect. I'm checking this one because I think it's a little bit harder than some of the other ones. So good. Good job. So what's your phenotype? If you have an A and a B, which is your genotype? What would your phenotype be? Because they're codominant, it would be AB. That person would have AB blood. So they would have points and squiggles on both. So here you just write AB, okay? So the next one we'll look at is chromosome number 1. That's going to tell you your rhesus factor, which is still part of the blood, okay? So find chromosome 1. Remember, Rh positive is the dominant. So first write down what the letters are, because that's your genotype. And then figure out whether you've got the phenotype is positive or the phenotype is negative. What happens if you have one that's Rh positive and one that's Rh negative? Well, come on in. We're just starting this activity here. So here's a paper for you, and you'll have to just share with somebody maybe over there that has already lined up the chromosomes. Oh, I need paper. Here's another one. Okay. All right, next step, did we all get that one, the rhesus factor? We're all good? Okay, so the next step we're going to look at chromosome number 1, and we're going to determine whether or not this person has the peak hairline on the forehead. Okay, so look at chromosome number 1. Do you find capital P, lowercase p, or one of each, or 2 of each?
How do you tell?
Yeah. Didn't I underline them? I think
No.
the capitals I underlined.
Oh, okay, and then mine are both lowercase.
Boy, that doesn't look underlined, does it? See, she's got it underlined.
Yeah.
So if it's not underlined, it's lowercase.
That's
You got an upper and a lower. Sorry.
It's alright.
So if the P is not underlined, it's lowercase. All right, and so if you have uppercase, that's the dominant. So if you have one uppercase P, your phenotype is going to be widow's peak. Okay, if you have 2 lowercase, it's no widow's peak. Okay, then we can look at chromosome number 4, and we can look for freckles. So you're going to see capital F or lowercase f. Actually look at his, okay, because you need to fill this out. I'll get your pencil. Okay, so if you have, so your phenotype, no freckles is the dominant. So if you have a capital F in there, that's no freckles. Okay, it's only 2 lowercase f's that are freckles. Remember we said dominant doesn't mean more prevalent. Okay, so in this case, no freckles is dominant.
Mine has freckles. It's cute.
Okay, the next one is dimples, and that's on chromosome 5. So you're looking for capital D's or lowercase d's. And so if you have one capital letter D, you have dimples. So far so good we are? Yeah? Okay. The next one, chromosome 6, is going to be cleft chin. So you're going to look for a capital N or a lowercase n. Okay, in this case, cleft chin is dominant. So if you have one capital N, at least one, you have cleft chin. Okay? Number 7, on chromosome number 7, is cystic fibrosis. So you're looking for a capital C or a lowercase c, and I underlined the capital C's. That's how you can tell, because they look the same too. Okay, so no cystic fibrosis is the dominant. So if you have 2 lowercase c's, that's cystic fibrosis.
Oh, cool.
Okay, on chromosome 11, we're looking for sickle cell anemia. So you're looking for capital S, which would be underlined, or lowercase s. So the dominant is no sickle cell anemia, so you would have to have 2 lowercase s's to have sickle cell anemia.
Oh, I get that.
Okay, on chromosome 15, we're looking for hitchhiker's thumb, which would be capital H or lowercase h is what you're looking for. no hitchhiker thumb is what's dominant. So if you have any capital H at all, you have no hitchhiker's thumb. So my husband has 2 lowercase h's because he has it. I must have at least one capital. Okay, so now we're going to look at chromosome 21, and we're going to count how many chromosomes there are. Did you get 2 chromosome 21s or 3?
2.
Okay, so 2 is not Down syndrome. 3 is Down syndrome, or trisomy 21. Okay, then we'll look at chromosome 22, and we're looking for a capital E or a lowercase e for attached or unattached earlobes. Okay, the dominant is to not have them attached. So if you have at least one capital ear, your lobes are unattached. I'm sorry, one capital E. If you have one capital E, your earlobes are not attached. Now here comes the sex-linked trait, color blindness. So we're going to look at chromosome X. And what you're looking for is the capital B or lowercase b. So it may be the case, like if you're male, you only have one letter, right? So if you have one letter that's a lowercase b, you have color blindness. If you have 2 letters that are lowercase b, you have color blindness. Everybody else has no color blindness. Are you guys familiar with that? That color blindness? Like often, there's an example over here on the poster that people that are colorblind would not be able to see the numbers inside of these. That's like how you do a colorblind test. Is can you read the number?
Yeah, my dad's colorblind.
Yeah, it's pretty common in males. So like we talked about. So those are all the different types of things. So what I'd like you to do now is one thing you can do is check your answers because I have the answers over here. The other thing I'd like you to do is go to somebody else's kit and read their kit, just like we did. Only, no, don't switch papers. You actually want to switch seats, but you want to make sure that you got the right answer too. So you can look over here at the keys and then read somebody else's. Just read one, and then we'll stop. How'd you do?
I don't know.
Awesome. How'd you do? Okay. That's okay, but what the phenotype means is actually what you see. So that's what phenotype means. And so what you would want to do is you would just put, this person has the peak, and this one has no freckles. That would be your phenotype, what you actually see.
Oh, okay.
Yeah. I don't lose my chromosomes. I'd never find them. Okay, so what we'll do is we'll talk a little bit about biotechnology, but we're also in the process going to talk about how DNA gets replicated, like specifically how that happens, and also how proteins are made. Okay? So we'll discuss DNA replication, transcription, and translation of RNA. So these are all new words. These are great to make like flashcards out of, you know, like what's this word? Let me test myself. Remember, a lot of science is if you know the definition, you know what it is. We're going to define biotechnology, some applications of biotechnology, DNA and sequence. We'll talk a little bit about the Human Genome Project, DNA fingerprinting, which is what they use in court cases. It's pretty much identical to paternity testing in a lot of ways. And then genotyping for health and ancestry, if we probably would do that next time. If we do it at all, we'll do it next time. Okay, so let's get reacquainted with DNA. So we know it's a double helix, and we know it's made out of 4 different nucleotide bases, A's and T's, just like peanut butter and jelly, C's and G's, cookies and milk, right? They go together. Every cell has a complete set of your DNA, no matter whether it's a muscle cell or it's another good thing, a skin cell. No matter what it is, they all have the same copy of DNA. But what makes them different is how they express their genetics. Okay, and so that's like, we probably won't get a lot into that today. But that's what makes things different. Is it's not only what you have, but what you're expressing. Okay, we have 99.9% of DNA is the same for all humans, and DNA essentially carries genes for your traits, but there's only a small amount that actually codes for something meaningful. A lot of it is not meaningful. They used to call it junk DNA, but they realized that it has functioned since then. But really, it's only a very small percentage that actually codes for anything to be made. So we also can see that gene expression changes with time. So I'm getting older. My face is changing kind of a little bit. I got some little wrinkly laugh lines. And, you know, you just change as you get older. I lost some of my freckles that I had when I was a kid. And that's all. It's not that my genes have changed, it's their expression has changed. So we know that DNA is found in the nucleus, and so these are just to remind us that we saw the nucleus when we did the plant cells and the cheek cells. So DNA, the genetic material, okay, so we just looked at the karyotype as like these chromosomes, right? Doesn't that shape look familiar to what you're just looking at? And so what you can see is like there's like the super coiling of DNA to fit in there. And then when we talk about prophase, when they're starting to coil up, you know, this is the stringy stuff is actually starting to come together into that kind of organization. And so this kind of just shows you how complex it is. It just keeps winding and winding, and then it winds upon itself, and that's how it gets so compact. So DNA replication. We're talking about mitosis here. Okay, DNA replicates itself. In order to do that, it needs an enzyme called DNA polymerase. What does an enzyme do? Tell me what an enzyme does. Huh? It acts as a catalyst. It lowers activation energy. It goes to work. Enzymes go to work. This enzyme is the builder. Okay? That's his job. He builds. So you'll see that in DNA replication, the DNA will split apart. This enzyme will come in and start filling up the other side because we already know what it's going to be. If I have an A, I know I'm going to put a T there, right? That's what he's going to do. He's going to put the matching letters, and then, so you'll go from one strand to 2 strands. From this, the whole idea of DNA is to make proteins. That's the whole function of it. So DNA is stuck in the nucleus. This is like a cell here. This is a cell, and then this is like the nucleus over here. The whole function of DNA is to make, ultimately, a protein. That's the whole function. So DNA cannot leave the nucleus. It's not allowed. It's against the law. But what it can do is make a little copy of a gene, which we call RNA, messenger RNA specifically, and that can leave the nucleus. So it floats around the cytoplasm until a ribosome grabs onto it, and then it starts reading it, right? So if this is the cookbook, this is a recipe, this is the dish, okay? So this one, this should be RNA polymerase. I'm sorry, this is a mistake. What I wanted to show you now is when you go from DNA to RNA, now you're talking about polymerase. Polymer is what? Many? Polymer, right? So polymerase is like making many. So this is RNA polymerase, and I use the light switch there because that means that gene is on. Something has turned that gene on and encouraged it to be expressed. So that's why that enzyme is going to go to work, make the RNA, messenger RNA, which is going to come out here into the cell. Okay, so these processes have some names. First is transcription. So scribe, right? Scribe, what does a scribe do? Don't they copy and write? Aren't they writers? Okay, so you can kind of think of that okay, we're writing down what DNA has to say, and we're going to put it in an RNA. It's going to leave the cell. What is translate? My husband speaks Spanish. Do I speak Spanish that good? No, I usually need a translator. So what does that mean? I need somebody to help me understand. Okay, so this is translation. This is the ribosome, and it's reading this, and it's going to end up with that product. Okay, a protein product. So that's kind of how you can remember what transcription and translation are. We have some videos to watch, but I'm going to move ahead so that we can actually start our next activity. So there's 3 types of RNA we're going to learn about. Okay, messenger, right? This is our recipe. The messenger is sending out the message to the cell. That's one type of RNA. Then you have another type called tRNA. This is transport RNA. So it's a little piece of RNA that fits right in the ribosome. It's really neat how ribosomes can change how they are based on what they're reading. And this will fit right in there. And it delivers the exact, these are the amino acids, 1 of 20, right? From cytochrome C, remember that? There's 20 amino acids. So this will deliver the correct amino acid onto the chain just because the ribosome will shift in its shape. The other type of RNA we have is ribosomal RNA. So this whole this whole molecule is made up of RNA. Okay, so 3 types. R for ribosome, T for transport, right? They're moving them around, and then M for message. What do you have to say? Okay. Okay, we're going to do assignment 3.05, but in a little bit. What I think I'd like to do next is go ahead and start the extraction. Alrighty. So what we're going to do is see, this is lab application now. This is what people do in labs, okay? We're going to look at DNA, and we're going to first pull it out of the nucleus. So we're going to extract it out of the cell, okay? Then we're going to need to make more of it. So if we were going to do something like a DNA fingerprint and use that in a crime court case, or if we wanted to do it to paternity testing, we're going to need a lot more than just a little bit to do that, okay? So our next job after we extract is to make more, okay? After we do that, we're going to cut it into pieces because that's where we get the individual's DNA fingerprint from. Everybody's pieces are different lengths. A couple of reasons for that. One is that there are repeating sequences that vary among us. It's like stuttering. It's like the DNA, the DNA, okay? So that makes the pieces shorter versus longer. And that's what we're looking at when we look at a DNA fingerprint or a paternity test. We're looking at the length of those pieces, okay? After we cut the DNA into pieces, we're going to need to separate the pieces out so that we can actually see what we have in that pattern, okay? So those are the 4 main steps. So let's start with our activity, where we're gonna actually start over in that corner, and this is going to involve you getting up to watch and participate. Procedure that also comes from Carolina Science. Would you be my banana peel, please? Go. Okay, and so what we're going to do is break it into some pieces and just throw it in. It's amazing how easy it is to extract DNA. Seriously, this is all stuff you probably have in your kitchen. Okay, so that's our first step. Our next step is to cover the banana with some salt solution. So I already made up some salt solution. And we're just going to fill this beaker up to the line. Just give it a little squirt in there. That's a black one, yeah. Oh, that's enough. Just enough. Perfect. Luckily, this isn't critical, so it's not going to affect anything. Okay, so now we have that in our beaker. Next step we're going to do is add 2 drops of dish detergent, which I have down there. And I am going to need somebody that has a phone with a timer. Does anybody have a phone with a timer? Okay, could you set it for 45 seconds, and would you put 2 little drops of dish detergent in there? Just little 1, 2. Oh, that's good. That's perfect. Perfect. Okay. So what we're going to do now is turn this on and off for the next 45 seconds. Are you all set? You tell me when to go, okay?
Okay.
Okay. Blender's getting old. It's time to stop? Nope. Okay. So he says that's 45 seconds. And so our next step is to pour some of this into the funnel. And basically I'm going to strain out all of the cell junk. Oops. So while we wait for this to get some liquid in there, let's talk about what we just did. What did we just do here? We took this banana and blended it. The physical action of the blender breaks the cell walls. Okay? We added salt. The salt disrupts proteins. It kind of untangles them, unforms them. The soap breaks down our cell membranes. Okay, so the soap's job is to break apart all of those lipids. And then I will be adding meat tenderizer. This is pretty interesting. What we're doing now is funneling out all of basically the cell physical parts. Okay, so what's left in there is all aqueous kind of solution, but there's still enzymes in there. There's DNA in there, and there's still other proteins that are protecting it. So I'm going to add a little meat tenderizer because that's going to disrupt that protein. And then after that, we'll be able to pull it up from the solution. Taking your time today. Awesome. get enough to have a couple of test tubes. I know, I'm going to put it in here. Okay, so we're going to add just a pinch of meat tenderizer. I guess we can do it that way. So like I said, this is going to disrupt more proteins, but these proteins are the ones that are around the DNA. So we're going to give that a swish. Okay. I'm just going to put some in the test tubes. I like to do at least 2 just to have like a second chance. Oops. I've got banana. Okay. Alrighty, and so now what we'll do is we'll talk about the next steps. Okay? So what we've done here is extracted DNA out of the cell. Our next step then is we need to make more. We need to make more because this is not going to be enough to do anything with. Okay? So here's how we're going to do it. We're going to cook it up in a pot. Here's my pot. I'm adding some DNA that I just extracted. Okay, that's the first thing that goes in. We add enzymes, the polymerase. Remember the polymerase goes to work? This is RNA polymerase from a specific bacteria. This is kind of cool. This is a bacteria that lives in hot springs in Yellowstone National Park. There's a picture of where you can see the snow is on the bank, but the spring itself is really hot. And it's very important that this enzyme withstands hot temperatures. Okay, so we're going to add some enzymes in there. Tac polymerase. We're going to add, well, if we're going to make up more DNA, don't we need more nucleotide bases? We need that. So here's some G's. Here's some C's. We're going to add some A's. Here's some T's. We're going to add a pinch of salt. Seriously, they add like salt. It's not table salt, but it's salt. Okay, and so its function is to sort of like stabilize the reactions. We're going to add a little bit of, I'll call it water because we're making DNA soup, but it's actually a buffer. It's just a chemical liquid that kind of lets things happen. And then we're going to add like a little spice, but we're going to call these primers. So if you want a specific piece of DNA made, what you're going to do is throw in the start piece. Here's where I want it to start, and here's where I want it to finish. Okay, because there's no need to copy the entire thing, right? Why would we copy the entire thing? That would be wasteful. But if we give it a start piece and a stop piece, that little polymerase is going to be able to grab on. It's going to know, because this little piece will bind onto our DNA when it comes apart. You'll see that in a second. And then that's where the little enzyme will know to grab on right there and start building. Okay? So what we do in the pot is we change the temperature. We go from hot to cool to warm. Okay, here's my DNA strands. My arms are the DNA strands. Hot pulls it apart. Cool makes the primers grab on. And then warm lets it build. Okay, warm is the builder. Okay, is when it's building. Okay, so what we're going to do is play sort of like a game here. Will you be my partner for a second? Okay, the girls are going to go hot. The boys are going to go cool. And we're going to go warm. Okay, ready? Girls say hot.
Hot.
Boys cool. Warm.
Warm.
So we just made 2 copies of DNA. Okay, girls.
Hot.
Boys. Warm. So we just made 4 copies of DNA because those 2 copies made copies. Girls.
Hot.
Boys. Warm. So we just made 8 copies. Okay, girls. One more time. Boys. Warm. So we just made 16 copies, okay? And so it goes from 16 to 32 to 64 to 128, 256. If you do this If you do this about 20 times, it's over 1 million pieces of DNA of exactly the segment you wanted. Okay, so now we've extracted the DNA, and then we've made more. Okay, we're ready for our next step. Okay, so we have now all this DNA that we made. This is like a gigantic test tube. Okay, we're going to pretend. We have all of this DNA we just made from that reaction. That was called PCR, polymerase chain reaction, in case you've heard that from Dad. Okay, so we have all of this DNA. Let's throw it in the test tube. Now we're going to get another enzyme. What do enzymes do? They work. Right, they go to work. Act as catalysts. Yes, this one has a job. Instead of a builder, he's a cutter. Okay, he cuts. Every time he sees the same sequence of DNA letters, he says, oh, there's my sequence, cut. I'm going to cut it right there, okay, always. So it's like in this example, I'm just using a really simplified example, but let's say this is the sequence. Oh, sorry, you can't see. Let's see, this is the sequence, and every time you see 4 T's, let's say, it's going to say, oh, that's my chance to cut. Okay, so here's 4 T's, it's going to cut. And then you'll have a piece here, and you'll have all this piece. Here's 4 T's, I'm going to cut. Right, and so there's a break here, and this will be another piece. Here's 4 T's, I'm going to cut. And so what ends up happening is you have different strands of different lengths. From originally it was this long, now we have pieces from that one, 4 pieces that came out of that one. Making sense so far?
Yeah.
Awesome. Okay. So here is our enzyme. His job is to cut. We're going to just throw it in the test tube and let it sit for a while. Okay, and then when we're done, we're going to have a mess of DNA. Some are long, some are short, and 2 are in the middle. Okay, so now we have made more, and now we have cut. We've extracted, we've made more, we've cut. Now we need to separate our pieces out. Okay, so we're going to slide down to this next one over here. The real deal breaks, like really easy.
I'd break it.
Super easy. But that's okay. That's why these are fake learning ones. Okay, so what we'll do is we'll have something called a gel, and it's really just sort of like this semi kind of solid matrix. Okay, and so it has spaces in between it. And so little pieces of DNA that are tiny, they can boogie on through those pieces, up and down and over and under, and I'm all over the place. And so what happens is if you put a bunch of DNA in what they call a well, and you add a charge, okay, because DNA is actually negative, so it's going to move to the positive end. A little piece can boogie on through, it's going to go all the way to the bottom because it's a quick mover, right? Does that make sense? Big baggage, I'm a big old piece of fragment, I got all this junk, it's like, oh, climb over this one, oh, in that space, right? And so it doesn't go very far. Okay, and that's how you end up with bands. So this is what our little gel is like, and it's in a box of liquid, and here you can see they've added a current. We've put our pile of DNA in a well. It's because it's negative, it's going to move towards the positive, and the smaller pieces are going to move faster. And so what you end up in real life, it looks kind of like that. But in a nice, easy picture, it's like this. Okay. So what I would like actually for you guys to do is to practice loading a gel. That's what we call it. So here is your sample of DNA, and I want you to try to feel around for the gel, for the well. This is tricky, and it actually is tricky in real life because you want to get it into the well, but you don't want the air to get in too much because it'll make it spill all over. Can you see how in there you can actually see where it's been loaded?
Yeah.
So that's what I'd like you guys to do. I'd like you to try to feel how that feels, okay? So there's 4 different ones, and I have more dye here because I don't know if that's enough. And then fill the wells up, and then we'll just clean them out, and the next person. So fill them all up. Get a feel for it. Okay. Okay. And do the second assignment. Yeah, okay.
What do you want me to do now? I did.
Oh, so we're gonna let me get all these bottles out of the sink too. I'm sorry.
All right.
Oh, I'm sorry, I'm making all this noise. Actually, I think I'll be okay because I actually was hoping to get a little bit. Does it look like a test tube?
And that is?
The actual stringing of the DNA. I'll get down close to the ground and okay, let's cross our fingers and hope this is cold enough. This is just alcohol.
Do you need ice in this car?
No, it's see how it's floating? You can actually
Yeah.
see it floating.
Yeah.
That looks pretty nice.
Oh, that's what he said, didn't he? On the movie?
Which movie was that?
The one that we saw? Yeah, he mixed it up and the DNA
Yes.
was floating.
Who, Chris, Miss Witsek, could you take a photo, some photos of this? Because
Oh, sure.
I gotta try this one because you can actually see it, and then I'm gonna try
in
to
front of you? Is it
look
okay
on
if
the
I go-
side. Part
Around
the
that way?
C's, students. Part the C's. Miss Witsek is gonna get a
Take it.
photo.
She's got the camera right there. Right there.
Okay. Thank you.
Did you get it?
Yeah.
Now let's see what happens if I can actually string it up.
Oh, that's cool.
So this is all DNA. It's pure DNA because it solidifies in cold alcohol.
That's cool.
That is so cool. I want to do that.
Oh, I'm gonna do that one.
Well, you can do it here. You want to play with it?
Yeah.
Yeah, go ahead. I got another bottle. Let's see if I can get that. I only have one loop, but
That's so cool.
The DNA.
That's the DNA.
of the banana.
Oh, it's a banana. I've seen the inside of a banana. So cool.
You guys want to I don't know. Let me cork it, and then you can. That'll be safer. Let's do that. And so you can see it floating around in there.
Cool.
Yeah, I think so. I love
So can we
genetics and stuff.
Duplicate?
You could if you put it in the soup and added some primers and
So we
had the polymerase.
Bananas?
Well, that would require like tissue culture and media and stuff, but
So-
given the right scientific stuff, you could.
What if I did that for like a science fair?
That's
Would
what
I
we're all
win?
made
You
of,
think?
guys, right?
I don't know
Think I
how
could.
easy it would be to get some of that stuff. It would be, you'd have to, the right combination of hormones and the right combination of nutrients in the agar. I mean, that's tough.
What if I'm like a genius?
Well, you would get my vote, that's for sure.
Alright, I don't know what to do with this now.
Anybody else want to play with it? She wants to play with it.
Good for it.
Yeah.
Yeah, unless you get bananas that are from like Africa, you have some poisonous spiders in them.
Well, you're
I
gonna,
like see it.
there's a whole clump of it.
No. They have spiders in Africa that
I'm gonna delete it, okay?
Can I get that phone?
I have another
are, yeah,
one.
there's a way you can feel it and spiders are coming
That's
out.
my worst fear.
Really cool.
Kind of looks like a mucus.
Who wants to be the yearbook shot?
He's thinking of jellyfish, and you're like,
Want to be the yearbook
Yes,
shot?
I'm
Doesn't
hoping
it
it.
look like someone like, that's a movie? They float.
Someone spit into some water.
Yeah.
Come on, let's do it. You want to do a group? Anybody that isn't opposed to being in a picture, let's do a group shot with some DNA.
Yeah, right.
Can you see my DNA?
I can't see everybody's faces. If you can't see me, I can't see you. Okay. 1, 2.
Y'all look really
Steven, you
tall.
want to be in it? You got to move on the other side. I got her, but okay. Cool. All right. Ready? 1, 2, 3. Say DNA.